Methods of sterilization for inoculation needle, glassware, plant material, and soil. First of all, what is sterilization? Sterilization is a process in which we make something free from contamination. There are different methods of sterilization: physical methods, chemical methods, moist heat, dry heat. Uh, Autoclave is the best example of moist heat sterilization, in which we sterilize materials under moist heat. And for dry heat, we use oven. And physical methods, we use disinfectants like Dettol's and hypochlorite solution and many other solutions. Chemical methods, we use chemicals for sterilization process. Now, in this experiment, what are the materials required? Glassware, inoculation needle, plant materials. In plant materials, you may take seeds, leaves, small plant cuttings, soil, garden or potting soil, equipment, autoclave, hot air oven, Bunsen burner, laminar flow, sterile containers which are free from any contamination, disinfectant, sodium hypochlorite solution, example hydrogen peroxide solution, media. This is the medium which is required for the growth of microbes, nutrient agar for bacteria, and PDA for fungi, plant tissue, culture media. Sterilization of glassware. For the glassware sterilization, what you have to do, firstly put your glassware, firstly clean glassware thoroughly with detergent and water, rinse it with distilled water, wrap glassware in autoclave safe paper or aluminium foil, load glassware into autoclave, set autoclave to 121 degrees Celsius temperature and 15 psi pressure. For 15 to 20 minutes, allow autoclave to cool down completely before its removal. You can also use dry heat sterilization for the sterilization of glassware. Clean glassware thoroughly with detergent and water, rinse it with distilled water. Wrap glassware in aluminium foil, place glassware in hot air oven, set oven to 160 to 180 degrees Celsius. If in Fahrenheit 320 to 356 degree Fahrenheit for 1 to 2 hours, allow oven to cool down completely before removing glassware. Sterilization of inoculation needle. Hold the inoculation needle in a Bunsen burner flame until it glows red hot. Red hot. Allow the needle to cool down before use autoclaving. Wrap inoculation needle in autoclave safe paper. Autoclave the needle as described above for glassware. Sterilization of plant material. For plant material sterilization, we have to go for surface sterilization. Prepare a disinfectant solution, 1% hypochlorite solution or 3% hydrogen peroxide solution. Immerse plant material completely in the disinfectant solution for about 5 to 10 minutes. Then rinse it thoroughly with distilled water. Dry plant material on sterile filter paper. Media sterilization for plant tissue culture. Media sterilization means the culture media need to be sterilized before the growth of microbes into it. Prepare plant tissue culture media according to recipe. Dispense media into sterile container. Autoclave this media for sterilization. Sterilization of soil. Remove any debris from the soil. Loosen it. Place it in autoclave in safe bags or container. Autoclave soil at 121 degrees Celsius for 30 to 60 minutes. Cool it down. Moist it slightly. Heat it in oven at 160 to 80 degrees Celsius for 30 minutes. Allow it to cool down completely before use. Observation and result. After sterilization, examine glassware for any sign of contamination. Observe growth of microbes on media inoculated with plant materials or soil before and after sterilization. Safety precautions. Always wear appropriate personal protective equipment, gloves, lab coat when handling microbes. Use caution when working with autoclave, hot air oven and Bunsen burner as they can burn you. Dispose of contaminated material properly according to laboratory guidelines. This is all about the sterilization of glassware, plant material and soil. For glassware, we used autoclaving and hot air oven. For inoculation needle, firstly we flamed the inoculation needle until it become red hot. Then we autoclave it. For plant material, we went for surface sterilization. We immersed the plant material into disinfectant solution, 1% hypochlorite solution, 3% hydrogen peroxide solution. Then we sterilized the media. Then in this way we sterilized the glassware, inoculation needle and soil. That's all about this experiment. Thank you for watching.